Hello and welcome everyone to this podcast. We're going to talk about the cell cycle, its regulation, and cancer. And though we've written it about 100 times, we're going to write it again. Here's our cell cycle. G1, S, G2, and then M phase. We haven't talked a lot about the regulation of this cell cycle. We've talked about what happens at each part. Remember, during G1, we have the cell growing, getting ready to enter S phase. We mentioned it checks for mutations and all that to make sure that you don't copy mutated cells, or mutated genes, I should say. And then you enter G2, and G2 makes sure all that DNA that was copied, there's no mutations, and all the DNA has been copied. And then during M phase, those copied chromatids now get segregated into new cells. As we said in the last podcast, when cancer starts, it starts due to a single mutation that is typically found in the cell cycle to allow this cell cycle to grow out of control. And so I think it's appropriate that we talk about the cell cycle checkpoints. And we have three that we should talk about. We have one here, and so we're going to call this the G1 S checkpoint. At this checkpoint, the cells check to see if the DNA within them has any mutations. And the cell will stop here and will not proceed to the S phase until that's been verified. You then copy the DNA, and after you've copied the DNA, there's another checkpoint here. So this is our second one. And this is the G2 M checkpoint. At this point, the cell checks to make sure all the DNA has been copied, that there's no mutations, and it proceeds. And then our final checkpoint is here, our third one. I'm going to write it here because I don't have room up here to write it. And this is the M checkpoint. In this checkpoint, the cell makes sure that all of the chromosomes are attached to a spindle. This is important because if it's not attached to the spindle, the cell doesn't want to move forward, because by going forward, it's not going to segregate those chromosomes in the way it needs to. Once the cell cycle has hit one of these checkpoints, and it's been determined that either there's mutations in G1 or G2 that need to be fixed, or the spindles haven't appropriately attached, the cell does a couple of things, one of two things here. It will either fix the problem, meaning it will either repair those mutations, or it will attach, make sure the spindles are attached. And it can wait until that is accomplished. However, there are times when maybe there are too many mutations, or there's too much damage in the cells, or it's unable to attach the spindles. And that's when the cell will undergo what is known as apoptosis. Programmed cell death. The cells decide, so to speak, that the problems are too great to overcome, and the cell is programmed to die at that point. And this turns out to be a really good thing, because if it didn't die at this point here, it would proceed and pass along these mutations to the next generation. So it either fixes them or it dies. So any mutations in these checkpoints here will remove that ability to check for mutation. So if a mutation exists in G1S checkpoint, and you remove it because there's a mutation in it, right? Now, when a mutation is detected, it doesn't stop to fix the problem or enter apoptosis. It just immediately proceeds to S phase. And now you've just copied that mutation. And then you move into G2, and that mutation is still copied, and it proceeds. And now you keep doing this. You never stop at G1, and so it constantly bypasses this stop sign, accumulating more and more and more mutations. So there has to be like an appropriate brake to the cell cycle and a gas pedal to the cell cycle. We want the cell cycle to move forward when it's time to move forward, but we want it to stop when it's time to stop. So what are these two classes of genes, the brakes and the gas pedal, so to speak? Many genes that are linked to cancer can either be called tumor suppressor genes or proto-oncogenes. It's important to note that both of these genes are genes that we have in all of our cells, and they're doing a very important job. The tumor suppressor genes, these serve as the cell cycle brakes. They are the ones that stop the cell cycle at these appropriate times, and so we want them around. These are good things. It's just that when they are mutated, we remove the stop signs, and that's when a tumor suppressor gene can now lead to cancer. Proto-oncogenes are the cell cycles gas, so to speak. It's what moves it forward. So we can say moves forward, the cell cycle that is, and as the tumor suppressor genes, they stop it. Again, these proto-oncogenes are in our cells, and we want these genes to be there because we need the cell cycle to progress. Otherwise our cells don't divide and we don't grow and we don't survive. So these turn out to be incredibly important genes. However, when they are mutated, that's when the problems come in, because now you cause it's kind of like if you're driving a car and you have your gas down just enough to go at the appropriate speed, but then if you push the gas all the way to the floor, now you're starting to go too fast and it's harder to stop. That's kind of what's happening here in this cell cycle, that the gas pedal is pushed too far and now we're moving through the cell cycle at a rate that is harder to control even with a tumor suppressor gene. So I'm going to first spend a little time talking about these proto-oncogenes, and then we'll talk about tumor suppressor genes. Let's go ahead and erase this, and let's write proto-oncogene here. This is the unmutated name of the gene. It causes the cell cycle to move forward, as we said before. And then we need to talk about what an oncogene is. An oncogene is the same gene as the proto-oncogene, but now it's been mutated. And it forces the cell cycle to move forward without regulation. It's going too fast, so to speak. Oncogenes, that is going from a proto-oncogene to an oncogene, we usually think of this as a dominant mutation. And what we mean by a dominant mutation is that only one copy of the two alleles needs to be mutated. And now I want to talk a little bit more specifically about how a proto-oncogene becomes an oncogene. And I think to explain this best, it's good to look at a specific example. And I want to look at a signal that tells the cell to move forward with the cell cycle. So I want to talk about a situation where the proto-oncogene usually functions in controlling the cell cycle. So let's draw a cell here. And for whatever reason, the cell cycle right now is off. Maybe there's a mutation, maybe it's just not time to grow. For whatever reason, it's off, and so the cell's not dividing. And let's draw the nucleus in here. So that's our nucleus. And let's put some DNA in there, because we're all about the DNA. And there's a gene here that needs to be turned on in order for the cell cycle to move forward. Essentially, this is a proto-oncogene. Once it's expressed, the cell cycle will move forward. And the way it's going to be expressed is by having it activated. And the way this particular one's going to be activated is through a receptor. So let's go ahead and draw our cell over here, again with our nucleus. and our DNA, and with that same proto-oncogene here. And now what's normally going to happen is that we will have some sort of growth factor added. So let's just write GF for growth factor. We're going to have our receptor here, and this growth factor is going to bind to this receptor. When it binds to this receptor, it's going to go through a conformational change because upon binding it gets phosphorylated. The phosphate group gets added to it. Now we don't need to worry about all these downstream proteins that are affected, but there are many proteins that are affected by this phosphorylation event, and they are all activated. But the one I do want you to focus in on is this one called RAS. RAS. Okay, so now all of these proteins in this pathway are activated, and again, you don't need to worry about them, but what I want you to focus on is this one right here, this RAS, R-A-S. And RAS, when it's active, and this process here will activate it, it binds GTP. And as long as GTP is bound to RAS, it remains active. In its active state, RAS will activate various other proteins that are our direct transcription factors that will now enter into the nucleus, bind to this proto-oncogene, and turn this proto-oncogene on to drive the cell cycle forward. Now, any one of these proteins in this pathway are involved in driving the cell cycle forward. But I want to focus on RAS. How is RAS a proto-oncogene? How could we get a mutation in RAS that allows the cell cycle, or this pathway, to always be on and to allow the cell cycle to always be on? What I want to do here is draw the RAS protein and show you how a mutation could occur. Remember, I said when RAS is bound to GTP, it's active. So this is active. When RAS is bound to GDP, meaning it's been hydrolyzed, it's inactive. And then it becomes active again by exchanging the GDP for GTP. So how do you allow this RAS protein that's bound to GTP to always be active? Turns out you can get a mutation here in this structure here, the active site that binds GTP. Specific mutations can make it so that it always is bound to GTP, meaning that the GTP can never be hydrolyzed. And if the GTP can never be hydrolyzed, RAS is always active. As long as RAS is always active, it's always going to turn on this transcription factor, and then this transcription factor is always going to turn on this proto-oncogene, and the cell cycle is always going to be on and making new cells. Again, a mutation in this GTP active site, where GTP bounds, prevents it from being hydrolyzed. If GTP cannot be hydrolyzed, RAS remains active all the time, and if RAS is always active, it's always going to activate these transcription factors, which bind to the DNA here, the proto-oncogene, and drive it forward, meaning driving the cell cycle forward. It's always going to be on, regardless of what's happening here. So that's an important point I should stress. It doesn't matter if the growth factor has bound in that case if you have the mutant form. You wouldn't have to have this here because RAS is always active. You've made these upstream players here obsolete. RAS is active because it doesn't hydrolyze GTP, always going to turn this pathway on. The pathway always on, you're going to accumulate more mutations, the cells are going to grow faster, they're going to outcompete the other cells, and that can lead to cancer. I began that discussion of RAS saying I was going to tell you the 3 ways a proto-oncogene can be converted into an oncogene. So the first one is a missense mutation. And our last example of RAS is a perfect example. How a single amino acid change on RAS allows it to always be able to bind GTP and always be active. So just changing that protein structure a little can lead to an appropriate change that allows that proto-oncogene to be converted into an oncogene. Another way is by gene amplification. So how does gene amplification work? Well, if you have one gene that's a proto-oncogene doing its job, regulating the cell cycle, but if you amplify that by generating more than one copy of that gene, now you're increasing the total amount of proto-oncogenes being produced. And now you will have an oncogene. So gene amplification is when you have more than one copy of a proto-oncogene. And that extra copy can drive the cell cycle forward. So if we draw our cell cycle here, where we have G1, S, G2, and then M. Let's say there is a proto-oncogene that functions right here at the G1S checkpoint. However, in its single copy, it's regulated. It's able to keep the amount of this proto-oncogene in check so that the cell cycle doesn't move forward until the brakes are released, until the tumor suppressor genes have created the situation where the cell cycle is ready to move forward. But if you say have 4 times the amount of proto-oncogenes because of gene amplification, by creating more of that gene in the cell, now you can overload the system. Now the cell is forced to move forward even though it's not ready, because you have made more proto-oncogenes than the cell is used to having. And so it can't combat it. The last one, I'm going to need a little bit more space to draw, so I'm going to erase this. And this is not going to be a new topic for us, but it bears repeating here. Chromosomal translocation. And so let's draw 2 chromosomes. And these are non-homologous chromosomes, so we're going to make them different sizes so it's clear we're not talking about the same chromosome. And let's say we have 2 genes here. Let's say this gene is a proto-oncogene on the green chromosome. It's going to have a promoter that is regulated. Meaning you don't always want to turn this promoter on. You only want to turn it on at the time when you want the cell cycle to grow. Let's say this other gene here, well, for now let's just call it a maintenance gene. And what I mean by that is that this gene is always turned on. Maybe not at a super high level, but it's always turned on because it's performing a function all the time. And so it's always on. So this promoter here, let's call it, let's go always on. Alright, so this promoter is always on. This gene here that I drew at this line here is always on because it's a general maintenance gene. Always has to be on. This one's highly regulated. Well, you could imagine a situation where we have a translocation now. A reciprocal translocation, where this green one now comes over here, where that proto-oncogene is, and the purple one from the first chromosome moves over to the green one. Now let's remember we had a gene right here from the green one, and then we had this maintenance gene from the purple one. Now what's happened is this maintenance gene here has this highly regulated promoter. And now, well, we'll come back to that in a moment. And now this proto-oncogene has this promoter in front of it that's always on. So what has happened because of this translocation event, in fact, let's make sure we label this translocation. Is that we've moved this proto-oncogene from a highly regulated promoter to one that's always on. So now what we're doing is we're always making proto-oncogene, always being expressed. And that's a recipe for disaster, because now you have created a situation, or the cell has created a situation, where it's always turning on a proto-oncogene. And since now there's less regulation of that proto-oncogene, now it becomes an oncogene. So an overexpressed proto-oncogene becomes an oncogene. Now sometimes, depending on what this other gene is that's been moved to the highly regulated promoter, it's not going to be turned on. And in some cases that can actually help the cancer out too, because this gene may be a gene that's important in the immune system. And now you place it under this highly regulated promoter and it might now be turned off. So you create a situation, or the cell has created a situation where it's always expressing the proto-oncogene and it's not expressing this maintenance gene that might be important in protecting the cell against this overgrowing population. And the last thing we want to talk about with oncogenes is the role that viruses can play. Let's write cancer viruses. Early in the day, when they were first starting to investigate cancer, they had just gotten done curing diseases like polio, and so they felt that maybe cancer was caused by a virus. And we have discovered that some cancers are, but most cancers aren't caused by a virus. Which at the time was probably a little disappointing, because we can make vaccines against viruses. So as it turns out, we can't make vaccines against most viruses. We'll talk in class, hopefully, about one such virus that we can make a vaccine against. And I want to talk about this in a fairly generic sense. So we have our human cell here, and then we could have a virus here. And it doesn't have to be human, I'm just writing human. It could be some other multicellular organism. But the virus can enter the cell, and once in the cell, it can produce an oncogene from its own genome. Now the virus may incorporate into the host genome, or it may not, depending on the virus. But this oncogene, somehow or another, activates the cell cycle. For instance, it could activate the protein that we talked about before. It could activate RAS, so that RAS is always active. Whatever it's doing, and there's several examples of this, it can introduce this oncogene that ultimately drives the cell cycle forward without regulation. And the advantage to the virus is that now you're growing the cell, or reproducing the cell, quickly, and as the cell grows quickly, the virus gets to replicate its genome. Now ultimately that can mean the death of the host here, but the virus's job at this point is to copy its genome many times. So this turns out to be an attractive way for the virus to accomplish this job. Unfortunately, it comes at a cost to the host. Okay, that's all I wanted to say about oncogenes. So now let's start talking about tumor suppressor genes. Now remember, the tumor suppressor genes are characterized as providing a stop for the cell cycle. Typically, these are recessive mutations. Meaning that both copies have to be mutated. Both alleles have to be mutated. As long as one good copy is there, one unmutated copy, that's usually enough to stop the cell cycle. Now I'm going to talk about some specific tumor suppressor genes. And namely, I want to talk about this pathway that uses P53, retinoblastoma protein, which we abbreviate RB, transcription factor E2F, and how they're going to function in this pathway to stop the cell cycle. Alright, so in talking about these tumor suppressor genes, so to explain tumor suppressor genes, I want to draw a large nucleus here. And in this large nucleus, I'm going to draw some DNA. In this DNA, we have a gene. And this gene codes for an enzyme necessary for DNA replication. What it does during DNA replication isn't relevant for this conversation. It just is needed to replicate the DNA, so that means it's needed for entry into S phase. And so until the cell is ready to copy the DNA, there's this enzyme up here called retinoblastoma. We call it RB for short. It's a gene, well, this enzyme is coded by a gene that was first linked to a retinoblastoma, which is a childhood eye cancer. But this particular enzyme is found in all types of cells and plays a pretty important role as a tumor suppressor gene. So what retinoblastoma does is it holds on to this transcription factor called E2F. E2F, as long as it's bound to the tumor suppressor gene retinoblastoma, it can't transcribe this gene. So it keeps the gene off. And it's going to remain here until the cell has made sure all the DNA is mutant-free and is ready to enter the S phase. So in order to release the E2F, so we can move into the S phase, the retinoblastoma is phosphorylated by this enzyme called cyclin CDK. Cyclin CDK adds a phosphate group to retinoblastoma. That causes a conformational change, releasing retinoblastoma, has this phosphate group on it, and it releases E2F. And now E2F can bind to the DNA, to the promoter, turn this gene on for DNA replication, and now the cell cycle can proceed into S phase. And it'll remain like that until it's necessary to stop again. Once all the DNA is copied, this phosphate group is removed by another enzyme, and E2F is once again bound by retinoblastoma. So as long as RB is hanging around here, is able to do its job, it's going to help regulate whether or not the cell enters into the S phase. So it's an appropriate break to the cell cycle. It is a tumor suppressor gene. Next, I want to talk about P53's role in this. And P53 is known as the master regulator of tumor suppressor genes. And so I want to draw over here what P53 is doing. Okay, what I want to do now is draw another part of this nucleus. Now these two are still the same nucleus. I'm just drawing it in two separate circles so we can talk about two separate pathways, all within the same nucleus. But what P53 will do is it will look for DNA damage. So here's some DNA, and let's say it's damaged, and I'm just going to put an X, and I'll just put an X here to signify that it's damaged. P53 now will, this is P53, bind to the damaged DNA, and it will do one of two things. If it's too much damage, it will target the cell for apoptosis. That is cell death. A good example of this is after you have a sunburn and your skin starts to peel. That's an example where P53 has noticed so much damage in the DNA that it's better to kill those cells off than to pass those mutations on. It can't fix that many mutations. So the cells die, and in that case the skin peels away. However, if it's going to fix it, it needs to stop the cell cycle. It needs to cause retinoblastoma to bind to E2F. And the way it does this is by activating this gene called P21. I know, another gene. But P21 is this protein and its job is to inhibit cyclin CDK. So I'm going to say this again. If the damage is going to be fixed, p53 signals the cell to stop the cell cycle so that it can fix this DNA. So in order to stop the cell cycle, it activates this protein called p21. p21, now this is all in the same nucleus, so to speak here. p21's job is to bind cyclin CDK and inhibit it. It doesn't destroy it, it just inhibits it. And by inhibiting it, it cannot phosphorylate retinoblastoma. So retinoblastoma will remain in the bound state with E2F. E2F cannot bind to the DNA or transcribe this gene necessary for replication. So again, p53 makes p21 because it wants to stop the cell cycle. An effective way to stop the cell cycle is to block cyclin CDK. If you block cyclin CDK, it cannot phosphorylate retinoblastoma. By doing so, it's always going to be remain bound to E2F. As long as it's holding on to E2F, you're stopping the cell cycle. Once that DNA has been repaired, p21 will be degraded, and once it's degraded, it's no longer inhibiting cyclin CDK, and now cyclin CDK can phosphorylate retinoblastoma, releasing E2F to bind down here, turning on the gene. So you should be very familiar with this pathway, and these three situations include no DNA damage. What are all the proteins doing? 2, with DNA damage. And then think about this situation in the case if p53 was mutant, or if retinoblastoma was mutant. So think about mutants. How could some of these mutations in retinoblastoma, p53, lead to cancer? Also p21 could lead to cancer. So think about how those mutations in those tumor suppressor genes, particularly retinoblastoma, p53, and p21, how those mutations could lead to cancer. Alright, that's all I have for this podcast. So as a brief summary, we covered a lot here, so I'm not going to repeat it all, but as a brief summary, you should be familiar with oncogenes and their normal counterpart, proto-oncogenes. And how proto-oncogenes can become oncogenes. We talked about 3 different ways, and we talked about the RAS pathway quite a bit. You should be familiar with tumor suppressor genes, what they normally do in the cell cycle. You should pay particular attention to p53, retinoblastoma, and p21. These are the tumor suppressor genes and how they work with the CDK cyclin complex to regulate transcription, and how it keeps the particular cell at the G1S stop point. You should be familiar with what these different genes are doing in their mutated state and non-mutated states. You should be familiar with the differences between oncogenes and tumor suppressor genes. If you have any questions at all, please make sure you come talk to me. If not, I'll see you in class. Bye.